25 minutes a bit about what we here in Celsi and in Singapore are doing with this relatively new tool of wastewater-based surveillance, wastewater surveillance, and how, like, what in our opinion here could be the next steps for this method, and yeah, how it can be generally improved. So, okay. So maybe some facts about Singapore, because maybe like me, you might have not heard about this before. You might have transited away to Australia or something. So Singapore is a pretty tiny island nation at the most southern tip of Malaysia. It has around 130 square kilometers. Sorry, I don't know how many miles this is. And since the 1950s, it increased by around 23% due to land reclamation. So the island is really giving its best to grow a bit. It has 5.7 million inhabitants, which might not sound too much, but considering its size, this is the third highest population density in the world. As an island surrounded by ocean and Malaysia and Indonesia, it has a pretty particular drinking water provision, which is the 4 national taps: imported groundwater, urban reclamation, reclaimed sewage, reclaimed wastewater, and desalinated seawater. So of course we are talking about wastewater, but you might understand that in such a small scale environment, everything in the water cycle is very immediate and very much close together. So Singapore has 4 different wastewater treatment plants. 3 on the left side, or on the left side of the island, are basically catering to most of the industry and to some of the newer developments, while the right side, the one next to Changi International Airport, is treating about 1/3 of the population, more than 1.5 million population equivalents. So we right now receive wastewater samples from all these 3 wastewater treatment plants, and with a team of maybe like 10 people are pretty much able to follow, to trace the transmission trends of the virus in all of Singapore. So if you compare this to a clinical approach, you would need considerably more workforce and also money in hand. What we are also doing, we are here in the very right side of the island at Nanyang Technological University, and this university has a pretty nice campus. It was established in the 80s, but especially because Singapore, as you could imagine, pretty high real estate prices. So the around 31,000 students tend to live on campus if they can, because for them it's the first time moving out, living outside of their home apartment with their parents. So all year round we have a pretty high tendency of the students to stay on campus in a pandemic situation like with COVID. Of course, these high density living spaces tend to be the main driver in the pandemic transmission trend. So this is why you're here. As you might have heard, wastewater bears a lot of information about not only the transmission trends, about local outbreaks, but can also be used to identify either the dynamics of one variant of concern, COVID variant of concern, overtaking the other, and to maybe even give you a bit of a better idea of how the pandemic trend in outbreak is behaving in any certain population. So what our, maybe you can see on the right side, what our collaborator at MIT found is that the virus numbers in the wastewater foreshadow the dynamics of clinical presentation by quite a bit. This was early in the pandemic when people were basically seeking medical attention for a lot of these symptoms that they might tend to ignore by now. So this is a new point that hasn't been added to this showcase of publications because there is not too much. But right now, even if you might have less of a cooperation in your population to seek out testing, like maybe if free testing stops, you can still be able to have a pretty good idea about how the virus is spreading by just looking at the wastewater. So maybe some of the limitations that we encountered during looking for SARS-CoV-2 in Singapore. So pretty early in the pandemic, it turned out that even though this is a respiratory virus, it infects people via the airborne route, and it mostly manifests in lung-associated, respiratory tract-associated symptoms, like what you found for the cold. It still reproduces in the human gastrointestinal tract. So coincidentally, we find quite a lot of SARS-CoV-2 RNA in the sewage. When you look at something in the sewage, in wastewater, there are certain factors that might influence here it says infectivity, but also the presence and the ability of ddPCR or Qpcr to quantify those viruses. So part is the water age between the point where virus enters the sewage stream and the point where you take a sample. There could be considerable distance, and of course, virus RNA, like all genome material, just tends to break apart at temperatures which are not suitable or not in their cardinal range. So if you want to draw some conclusions, you also need to know about the population in your sewer shed. You need to have a bit of an idea about the persistence of the virus under environmental conditions. So how long does it take for the virus to degrade due to influences like temperature, pH, grazing microbes. In sewage, of course, you find stuff like detergents and substances which might not be harmful to the virus, but which will inhibit the efficiency of the polymerase used for PCR, for ddPCR. Not so much an issue in Singapore, but in areas with a lot of agriculture. You also need to be aware of non-point pollutions, like maybe at a certain point, runoff from fields, from streets, from like lawn areas might add to the virus load in the sewage treatment system. So you might draw a conclusion based on a concentration of virus, which is not representing only the population in your sewer shed, in your area of investigation, but also coming from other sources. Not so much a case for SARS-CoV-2, but if you look at viruses which are also excreted by other mammals, this would be important. Yeah, and the decay rate. So all of this is a bit maybe too general, because for SARS-CoV-2, it has been shown that no infectious viruses are present in the human stool, even if excreted by highly contagious individuals. So the fecal oral route, which you find for stuff like rotavirus, for norovirus, is not really an issue for SARS-CoV-2. So for us, looking for the virus in sewage, in wastewater, is mostly about drawing conclusions about what is happening in the upstream area of your sampling point. So how do people infect each other up there? Not that this might be a source of infection. So this is maybe just completely redundant for the audience here. So if you look at the general system of PCR, you would have a gene region. Here it is DNA. So for SARS-CoV-2, you would have an additional step on the left here, where you take the RNA, turn it into complementary DNA, cDNA, which is then brought together with nucleotides, with the building blocks of the genome, with the DNA primers, which are the targeted primers, which are binding to an area of your target, which is indicative for either the presence or for the variant of concern, for a mutation that would allow you to identify if this is the Omicron variant, if this is Delta, and you have some polymerase. So you have this multi-tiered repetitive process where you increase the temperature, so the double-stranded DNA breaks apart, the primers bind to it, the polymerase identifies the primers, takes the nucleotides to construct new strands, which then go together, and then in the next step, the same thing happens again and again. So you have an exponential increase if you add a probe, like a fluorophore, which binds to the same newly developed DNA product. The ddPCR, the Qpcr, the laser of this machine will be able to identify if there's a change in the absorbance, if you have a signal of the target you are looking for. So for SARS-CoV-2, again, maybe this might be redundant, this might not be necessary. This is just a very, very simple, not even an organism, because organisms by definition need to be able to reproduce by themselves. The viruses are basically something in between. So yeah, SARS-CoV-2, RNA virus, is spike protein on the side, like those tulip-shaped things. And fun fact, so the RNA, the genome of SARS-CoV-2 is not group-read when it is reproduced inside the human cell, which leads to those high variant of concerns, because if this virus would be DNA-based, the proofreading function of the polymerase in the human cell would not introduce so many misreadings, misbuilt genome blocks. So the variant of concerns would be much less for a DNA virus like adenovirus. So this here is an overview of the genome of the SARS-CoV-2 virus, and you can see that it's pretty basic, it's pretty simple. So it only has the open reading frame 1A, 1B, and then the spike protein, which consists of actually the functional region of the gene, which is spike, the E gene, the M gene, and the nucleocapsid gene. And as you see by those boxes up there, early in the pandemic, there were different approaches to quantify SARS-CoV-2. The Chinese Centers of Disease Control and Prevention proposed something in the ORF1AB, open reading frame region, while the United States Centers of Disease Control and Disease, and then backed up by the Food and Drug, I forgot what the A is about. So they proposed a set of primers and probes and PCR protocols, which turned out to be used most like basically all over the world. So the US CDC primer set is right now what, if you use this, consider using this, you will be using. Okay, so if you go about quantifying virus in a sample like sewage, you will come to the point where you have a sample from your wastewater treatment plant, from your manhole, which is of course way too big to be analyzed by PCR. So you get something like 150 to 200 milliliter, and your PCR, ddPCR reaction will happen in 2 microliters. So in like thousands of dilution, like a factor more than like a huge factor in between those two numbers. So in environmental virology, there are certain approaches on how to maintain your virus signal while reducing the volume of your sample. And there's basically the question, how much money are you willing to invest? How good is your supply cycle? And how fast and how robust do you tend to work? So the centrifugal ultrafiltration, the first one in this line of viral concentration methods, uses a centrifuge filter unit, which is sold by quite some companies, has different filter membrane materials, which are more or less suitable to target RNA non-enveloped viruses like SARS-CoV-2. You can also use this very, very simple. The second from the left, PEG, polyethylene glycol, which is basically forming a polymer in your sample, which is entrapping, capturing the virus, and then you spin it down, and then you can remove it from the pellet on the bottom. And those ultra-negative filtration, ultracentrifugation, those don't really, it seems like they didn't really catch up much. So right now, basically, it's those ultrafiltration centrifugation units, or PEG, which both have their advantages and disadvantages, but which are giving you pretty comparable virus numbers in your follow-up quantification. So this is just like a bit of an overview. So early in the pandemic, people basically just looked at the wastewater treatment plant on the right side here. So they took a sample of the inflow, used their workflow to quantify the virus, and then they basically reported this number. Now it turned a bit more on the upstream region of this wastewater treatment plant. So university dorms, hospital and nursing homes, or just basically parts of a city, a village. You can just take a sample at a manhole at a convenient point where your population upstream is high enough to be meaningful, but not too high and not too mobile to that when you find that there's an outbreak, you don't reach the person or the people you want to reach. On the lower part here, you see something that is somehow or sometimes a bit forgotten. So if you have a certain virus concentration in your initial sample, which is or in your individual who's going to the toilet, this will basically, the initial concentration will go down along the workflow. So the loss of virus recovery between the person shedding it and your molecular quantification in the lab will be quite significant. So it's very important, if you consider doing this, this is what we also encountered, that you are very sure about your limit of detection, your limit of quantification, it is advised at a certain point to also look for a surrogate virus, which could be grass-phage, pepper-mild model virus. At the point of the RNA extraction, people started using murine hepatitis virus, murine norovirus, similar to SARS-CoV-2, but non-infectious to humans, which just tend to behave similarly in the treatment process. So by knowing how much recovery of your RNA standard of your surrogate you have in your initial sample, you might draw a conclusion about how much you lost and how high your basically hidden figure of your virus is in your catchment. Okay, so this is a bit about Singapore key dates, not too important. You just see that in the lowest of this, we had quite a bit of a first wave in early 2020, which then decreased. Then there was almost no COVID in Singapore for quite some time until first Delta, and then Omicron basically hit the community quite a lot, because at a certain point also the government opened up the country's borders, the flights started to pour in again, and we have a strong cross-border communication commuting to Malaysia. So at a certain point, people just started moving back and forth from these two countries. So I used to have one animation here of the hospital admission rate, because you might have heard Delta, Alpha, Beta variants were quite significant in their symptomatic. They led to quite some symptoms. They led to quite some mortality, morbidity. But with the vaccination rate, Singapore has like 94% 2 vaccination doses, 76% 3 vaccination doses. So people get sick, but they don't get so sick to be in ICU anymore. Yeah, so this is the same. Omicron took over the US, the same for Singapore. Very early utilization of wastewater-based surveillance in Singapore was worker dormitories, where workers were basically just put at different construction sites and then went back to their separate dormitories. So if someone's breath was contagious, transmitting the disease, of course, it was very likely to spread within different dormitories. Afterwards, we established the same method as the university campus. It was pretty nice because it helped us implementing in-person learning again after much earlier than if we would just do nasal swabs of all the students. Yeah. So what we found in this implementing is that it's very important to combine or to merge the laboratory and the policymaker side. Here in Nanyang Technological University, the administrative office needed to be very, very good in their communication of the virus rates, which would then lead to quarantine orders, visitation management. We needed to know about the occupancy, about the dormitories, about the poisons where we took the samples. We needed to know about the wastewater infrastructure when planning. Then we, of course, needed to tell the university administration about our sampling plan. We need to discuss it, our sample analysis, what are the limitations, and what do we take from this data. So this is not published right now, but it's under review. It should be available quite soon. So if you look at the pandemic flow over the past 2 years, it was pretty scary early on, and now it seems that everybody's starting to be rather okay with COVID and with people getting sick with COVID. So in parts of Europe, in Scandinavia, UK, I think also in the US, the clinical surveillance, the capacity that has been built up early in the pandemic starts to get reduced. So even though this is the gold standard, if you look at the virus load in an individual, of course, this has some limitations, some disadvantages, like clinical surveillance is pretty expensive. Healthcare workers get exposed to infectious virus materials, so you have a quite high incidence of SARS-CoV among healthcare professionals, not to be excluded that some of this comes from swabbing operations. So if you replace this with wastewater-based surveillance, this non-invasive method, you would be able to identify trends in your population. You can, be indicative for local clusters. You have very low cost per capita. It might be expensive to pay for all these materials, but if you count this down to a population of like 10,000 people, this gets considerably more cheap. Of course, no guidelines available right now, and you'd never be able to look at the individual infection, the individual being infectious. So maybe we will just rush through this because I want to make sure that Frank can also show. So after wastewater surveillance is used for SARS-CoV-2, after the pandemic is basically in check, it would be advised to keep the infrastructure, at least in parts, because like here in Singapore, we have a pretty high burden of disease associated with dengue, a virus that is transmitted by mosquitoes, and that leads to something which is pretty colorfully called the bone breaker fever. It is not necessarily deadly, but it is incredibly painful. And right now, if you want to check the incidence of dengue in your population, you either need to wait for people getting sick, and then it might be too late, or you need to look at the primary hosts, which are primates, which are some other animals like bats, or you could look at the vectors, the mosquitoes, and then you can look at symptomatic, asymptomatic people. Again, this is something in Singapore, you might be able to do this, but this is present all over the equator. If you look at lower resource settings, if someone comes into your healthcare center, says that he or she has fever and is just generally feeling bad, you might not identify this as something which, like dengue, which is highly transmissible. So it's good to be aware about the trends by using just some wastewater. If you do so, this is also not published yet, but almost there. So if you think about this, we have this nice table which can tell you about like what virus loads in wastewater do you need to get a based like how much wastewater sample you would have to process. So this will be around. This should be out in a few weeks. And I think the slides will be like we would share the presentation afterwards. Okay, so basically just what's next, the conclusion. So right now, one of the main questions was if people start getting vaccinated quite a lot, will this influence the shedding rate? So will the behavior of the virus in stool change because people started getting vaccinated? It doesn't look like it because the WHO approved vaccines are not attenuated live viruses like polio, which turned out in sewage afterwards. So likely that the shedding behavior will stay the same. Another question, will SARS-CoV-2 acquire the ability to infect people via the fecal-oral route? Again, this is very unlikely because it would be a huge shift in the virus behavior in general, but can never be completely excluded, and there could be another virus with pandemic potential that could infect. And then we would propose, based on our experience, that there should be standardization, maybe an ISO technical standard EPA method that would help implementing this. It would be advised to keep the infrastructure to be used for other viruses. And we shouldn't underestimate that this wastewater samples already collected can be used to identify future pandemic potential viruses, which are scarily called disease X associated viruses. So it would make sense to keep the infrastructure to be able to look for stuff in the future. Okay, so this is the theme. Thank you very much. And if you have any questions, I think the email, if you look for my name, the email should be available. So if you have any questions afterwards, just drop me an email. Okay, thank you. How are you doing? I think I'm done now. So if you could.
Done. I'm not sure how to start my slide. We had the same challenge in practice. It's not starting. It's not flipping over. No. Oh, maybe this is it. I think this is it. Hopefully everybody can see my screen. Mats, can you see my screen? Fabulous. All right. Well, thank you, everyone. My name is Francisco Frank Biswarn. I'm with the Digital Biology Group at Bio-Rad Laboratories, and we've been working on digital PCR for over a decade now. And we're pretty excited about the technology and its adoption in all sorts of different environments, including wastewater. So before digital PCR and droplet digital PCR mainly was used in wastewater testing, it had been used in the past for water monitoring in all sorts of different activities. So for example, monitoring invasive species, looking at Asian carp in the Great Lakes or in Mississippi, looking at invasive pythons in Florida, and also invasive shellfish in various bodies of water. So water testing is not new to this technology. It was also used to look at population density analysis. So if we wanted to know how many fish were present in a certain area, historically you would have to go fishing and pull out a certain number of fish in a certain amount of time and say this is the average speculated population. Well, unfortunately for fishermen, there's an easy way to do it, which is to collect environmental DNA and to monitor that. So this has been going on since early 2013 with droplet digital PCR, but more to the wastewater spectrum of things. As early as 2014, various groups, including the SCRPS group in California, we're looking at fetal indicators in wastewater downstream and its effects downstream mostly, instead of looking at upstream. But of course, with the advent of COVID, researchers started looking at the COVID-19 virus. Was it present in wastewater? Early in 2020, it was discovered it was. And since then, many labs have been using droplet digital PCR to accurately quantify and monitor whether the virus is increasing or decreasing as a function of time at a specific location. And of course, the idea behind this, as Mats has mentioned, is to be able to see patterns and predict what will happen in that environment. Do we need to isolate individuals? Do we need to increase the medical resources in a certain area? All of this to try to mitigate the situation, but also plan ahead. So over the last couple of years, we've seen droplet digital PCR being adopted in hundreds of labs all over the world. And what's interesting is initially, most of these labs were mostly academic facilities. But as time progressed, we've seen more and more applied labs, both at state health labs as well as wastewater facilities, start adopting this technology for monitoring, in this case, COVID. So why should somebody consider digital PCR for waterborne epidemiology instead of just Qpcr or PCR? Well, PCR is not going to give us a whole lot of information other than the sample or the target of interest is present there. But digital PCR and mostly droplet digital PCR has some unique features that really make it outstanding for analyzing low levels of DNA or RNA in a complex sample. And of course, I think most people will agree that sewer water and wastewater is a pretty mixed environment with all sorts of stuff. So some of the key advantages of digital PCR over other applications, of course, absolute quantification. So we can actually quantify the number of molecules that are present in the sample directly, and we're not dependent on a standard curve. And this is an important separation from what we often see in other techniques, is that generating a standard curve and maintaining those standards at the proper concentration so we can correctly say, okay, well, these CTs are equivalent to this amount of material, is a lot of work and is not always done correctly. So we can skip that part. We still need positive and negative controls. Controls are just good science, but we don't need a standard curve, which is really tremendous. We can also quantify very accurately down to plus or minus 10% of the actual sample, copies in the sample. So this is very, very high precision. Digital PCR in its nature is actually tolerant to a certain amount of sample inhibition. I mean, if you throw a rock in it, it's not going to work, but it is tolerant to some minor inhibition, which of course gives its accuracy a little bit more power. The workflow, very similar to Qpcr in that you prepare your master mix, you add your primers, your probe, some water, put in your template, and you run it in a thermal cycler and run. Now, what's really interesting about digital PCR is its reproducibility and inter-user variability. Reproducibility is very high, inter-user variability is very low, and this is because of the mechanics of the technology. And I'll explain a little bit more in a few slides. So the data is simple to analyze, and the results are actually objective as opposed to subjective. So it's pretty easy to analyze and to get the right number. So all of these parameters make digital PCR an ideal technology for Sentinel programs where we want to see, are there small changes over a certain period of time in a certain area, and so forth. So for those who are not familiar with digital PCR, you may have heard the term before, or you may think, okay, this is just like PCR. Well, it is and it isn't. So digital PCR basically is a combination of what we call direct molecular counting and distribution statistics. So what we typically do is we take what would be our Qpcr reaction, let's say 20 microliters, and we split that into 20,000 subreactions. So these are little droplets that are sub-nanoliter. And then we take these droplets and put them in a thermal cycler and amplify the template DNA in the presence of primers and probes. So if the target of interest is present, of course, just like with Qpcr, your probe will degrade and you will get a fluorescent signal. But what happens is we get the fluorescent signal in individual droplets. So that allows us to count the number of positive droplets, the number of negative droplets. And then we put in our good old equation here, which is Dr. Poisson's equation for small numbers. And in essence, we can accurately quantify the number of target molecules present in the sample. So it's pretty straightforward, pretty basic. This concept has actually been around longer than Qpcr, but it was not affordable at the time. So what does data actually look like? So if you look at this plot that I have here, you'll notice that there is on the y-axis a scale, which is the fluorescent signal. On the x-axis, we have the droplet. So in this case, we have about 18,000 droplets. And you notice that some of these little dots are high, some of them are low. The ones that are high had the target of interest in them during the PCR process, and therefore amplified and gave us a signal. The ones that are low basically didn't have a target of interest in them and gave us just a basal residual signal that our assay would have. So for analysis, what we simply do is we set a benchmark or a threshold, and this can be set automatically where by the computer, where these droplets are deemed positive, these are deemed negative, and we simply calculate the concentration using Poisson statistics. Now, if we do have inhibition in the sample, what typically happens is instead of having positive droplets that are way up high here, they tend to come down a little bit lower. But they're still positive as long as we set them, to be above this threshold. So that, in that sense, we are more tolerant and can deal with a certain amount of inhibition. Now, this analysis can be extended into multiple channels, multiple colors. So this is an example of a 4-target instrument where we see assays being analyzed in FAM for one target of interest, in HEX for a second target of interest, Sci5 for a third, and Sci5.5 for a fourth target of interest. And because of the partitioning process, there's actually very little competition for resources within all of these droplets. So it's not like with Qpcr where if I have one target of interest that is at high abundance and a second one is at low abundance, the high abundance one tends to monopolize all the reaction consumables, and I don't see the lower abundance one. The partitioning effect tends to nullify that, so we're able to see all our targets on all the different channels if they're there. We can also do some advanced design. So this is an example of one of our assays, our prevalence assay, where we're looking at the N2, the E, and we're also looking at a recovery target, in this case, mouse hepatitis virus, to see how much sample we're recovering. These can be combined with either singleplex or multiplex fecal indicator assays to give us a better picture of what we're seeing and what we're analyzing. And this can be done in a single well. Now, I mentioned that this technology is nice because of very low interoperator variability. And one of the key challenges whenever we're doing any science or any work that we're trying to compare one site to another site to a third site is intra-user variability. And this is an example that we ran 5 years ago. It's not using a wastewater sample. These are our blood samples where we were looking at a KRAS mutation. So this is an oncogene. Varies from the wild type by one base pair. And we sent, or some of our colleagues sent 6 study materials to 21 different labs all over the world to be analyzed. And out of those 21 labs, you can see the panel on the right, 18 out of 21 had, for all practical purposes, the exact same results at the different concentrations. The 3 labs that didn't analyze the data correctly, and when we're guided as to how to analyze it correctly, 2 out of 3 immediately said, oops, yep, this is what we should be doing. And at the end of the day, we had 20 out of the 21 labs that had the exact same results all over the world. So very reproducible, very powerful, and you know, not to be neglected, especially if we're trying to compare data from one facility to the next, one lab to the next, one user to the next. So Droplet digital PCR and Sentinel programs has some significant advantages, absolute concentrations at very low target of interest concentrations, not dependent on standard curves, tolerance to inhibition, and again, low inter-operator variability from person to person, site to site, lab to lab. And of course, the benefits of this include ease of use, stability of analysis, both longitudinally across time and across different labs, so that lessons learned in one area are transferable and translatable to other labs. This technology is cost effective, and of course the whole idea is to generate better data so that you guys can have better policy as to what should be done next. Now currently, and let's hope that this is soon to be a challenge of the past, we are focusing on COVID-19, but you know, let's knock on wood that this ends quickly, but we are also looking at other potential targets that could utilize this infrastructure that is already being put in place. So right now, COVID-19 is a big target, but there are labs looking at influenza, as Mats has mentioned, Zika, norovirus, adenovirus, Ebola, and we are generating assays for all of these. In fact, most of them are already available. I think the last 4 will be up sometime this week or next week. There are also a lot of publications that are looking at and present assays for rotavirus, zoonotic assays such as dengue and yellow fever and malaria and so forth, fecal indicators and so forth. So there are a lot of resources and tools available. And depending on where one is in the world, there are some targets that are going to be more interesting than others, even if there is no pandemic. And again, let's hope for that. So with that being said, I'd like to thank you for your attention. And if you, once this recording becomes available, if you follow the link on the bottom of the screen, it will send you to one of our websites that actually has information on not just digital PCR, but also getting funding from the federal government and other agencies for water-based epidemiology. So with that, again, thank you for your time, and I'd be happy to answer any question you may have in the time remaining. All right.
All right. If the audience has any questions, feel free to put those in the chat and chat those to everyone.
Oh, it looks like my presentation didn't go through. Oh.
Did everyone see the presentation pretty good?
So I saw the slide. Let's try to finish.
Oh, okay. Somebody, it looks like it's hit or miss. Not at all. Okay. Maybe it can be fixed in post editing. Post production, yes.
Yeah. Does anyone have any questions? Yeah, we got a couple of guys in the chat that say they saw it just fine. I think that was just an issue with Apple. I think it recorded and showed your slides, Frank.
Okay, fair enough. So I don't know if anybody has any questions, but I have a question for you, Mats. Recovery, obviously that's something that's important. And you were mentioning two methods, one which is a flocculation and the other one is a filtration. Which do you prefer under what circumstances and why?
So I had a bit of a side chat with Paul, who unfortunately couldn't see your slide, and he said that they are starting this on another island. So what we encountered is during the pandemic, when the flight bans were in place, it was quite difficult to get our consumables to Singapore. So PEG, the polyethylene glycol, it's
Mhm.
just so abundant in so many applications that even though it tends to co-concentrate the inhibition, so whenever there's something in the wastewater that interferes with the polymerase, PEG will most likely increase the concentration in your sample, even though, like, taking this into consideration, it was better for us
Mhm.
because it might take a little bit longer, but you are so much less reliant on supply chain issues.
Yes.
So yeah, also
Yes.
the cost factor. I think PEG is basically $20 a kilogram or something in this range, while those filter units are basically $25 a piece.
Hmm.
And different different applications are looking for the same unit, so you're always in competition with medical and with the industry.
Yes. Yep.
And Frank, I believe we have a question. The question from Michael is, does the epidemiology testing have the ability to distinguish between the different strains of COVID-19? And is there a lag in timing of reported test cases versus detection in the wastewater stream?
So I'll address the first part. Yes, there are assays specific to almost all of the variants. It's just a matter of getting the correct assay designed and made. We have a bunch of them available through Bio-Rad through our expert design. So, but you can also work with colleagues and other groups that have specific assays for them. So yes.
Yeah, we have our colleagues from the other university in Singapore, Wei Lin Lee, has an essay targeting different mutations in the different variants of concern. So yeah, they are open access. And maybe to the second part of your question, Michael. So funny enough, it has been shown that there's a time. So wastewater-based surveillance is very indiscriminate. So you just take a water sample, and regardless of what the population upstream of your sampling site thinks about the government, or thinks about getting tested, or thinks about reaching out to any healthcare facility, it doesn't matter. It will find the virus. And even though asymptomatic shedding is a bit lower than those people showing symptoms, it will still be captured and accounted for. So if you do wastewater-based surveillance, you will of course have the lag between taking your sample and getting the results from the PCR, which might be like 18 to 24 hours. But it has been shown in many parts of the world that during times of maybe when there's an outbreak in the healthcare facility that is doing the testing, in the laboratory, in the swabbing center, like sometimes those points, of course, also have COVID outbreaks. In case of holidays, like during 4th of July, during Thanksgiving, it just takes time. In Germany, there was a big delay in the holidays around Christmas. So people took samples or didn't go taking a swab because they wanted to see their families. So basically, you had 10 days between someone being relevant for your statistics and getting this result from this person. Wastewater-based surveillance, very indiscriminate. You just have it. You might need to know about your catchment. So how long does it take from each toilet to reach to your sampling point? But this can be found out with the facility, with the utility. So.
And the advantage is that it gives I guess the policymakers anywhere between a week, 2 weeks, and sometimes 3 weeks of a head notice that something may be coming down, depending on how quickly you catch it. So pretty powerful.
Awesome. And guys, I'm waiting on a few other questions, but if we don't have any, I would like to take the time to thank you, Mats and Frank, for a great presentation, and thank you to Biorad for sponsoring today's event. This concludes today's eShowcast, and I thank you for attending. The recording will
Thank
be
you.
available
Thank
at
you.
Oh, no worries, no worries. The recording will be available at wefbuyersguide.wef.org, and we will send our registrants an email tomorrow with this link. Please visit our event calendar to sign up for future events, and we thank you all once again for attending.
Great. Thank you. Take care, everybody.
Bye, everyone.